All right, well, if your head's starting to spin a little bit, that's okay. Take a break between lectures. But in the meantime, what I want you to think about now is why we should bother sampling. So I kind of explained before that one of the nice things about sampling is that we don't have to talk to every single member of a population in order to get good information, but I also told you there are some serious drawbacks to sampling incorrectly. We can have sample frame error. We can have a non-random sample where we can't project the results. We can also have something called sampling error, which we're not really going to talk about right now, but which can be a major problem. Well, the good news is that sampling works, and it works because of many of the statistical principles that are underneath it. And the great news is that if you know how to sample correctly, you can get some really great applications out of it without having to go to an awful lot of trouble. I'm going to offer you 3 examples, and these are all examples, by the way, that don't involve people. We're going to be talking about people in all of our other lectures, so let me just explain sampling to you in some other applications where it's very useful. So the first thing I want to talk about is medical tests. So let's imagine for a moment that you are feeling sick and you decide to go to the doctor. And you're going to, first of all, if you go to a doctor, you're usually going to talk to a nurse, and that nurse is going to take some basic measurements like your pulse and assessment of your breathing, your weight, and so forth, and then they're going to ask you to describe your symptoms. And once that process is complete, your doctor is going to come in and take additional measurements to see if he or she can qualitatively assess what's wrong. So to this point, a little bit of data has been collected, not a whole lot. Your doctor is really going to be focusing more on what you're saying and what you're describing about how you're feeling and make a qualitative assessment. Now, if your illness is common enough, that's probably all your doctor's going to need to do. But if it's not, your doctor is going to prescribe some tests. And the good news is that those tests are not going to require you to have to do an awful lot of work or to make your entire body available. Rather, what the doctor is going to do is have some blood drawn, maybe take a swab of the inside of your cheek, a sample of your bodily waste, or some other sample to send to the lab technicians instead of you. And these technicians are going to be able to use a bunch of different chemical processes to conduct a battery of tests on each specimen that they have to determine if you have any pathogens, bacteria, anything like that present in your body that might be making you sick. So think about this for a minute. The doctor just has to get little pieces of you, just a few drops of blood, a few cells from the inside of your cheek, some bodily waste that you didn't want anyway, and they're able to use even just little pieces of that to go through all these tests. It's pretty cool. And the reason that they're able to do that is because these can be assumed to be representative of the broader scope of what's going on in your body. These can be assumed to give a representative understanding of your entire body. So you don't have to go sit in an MRI and get scanned from head to toe and then have all of your blood pumped in and out of your body and have your entire digestive tract cleaned out, and examined and then put back into you. You don't have to go through any of that. Just little samples here and there can tell the doctor everything they need to know to figure out what kind of illness that you have. And in all these cases, that small sample is going to speak volumes about what's going on in your entire body. It's one of the neat things about medical science is that they're able to do that. And it's all based on the same principles that we're talking about here. Okay, so now let's think about air pollution. Think back to your high school or freshman level science classes, and you're probably going to remember that the air that we breathe is partially oxygen and it's also made up of some other gases. As you'll probably recall, gases are elements. They're in an excited state, and they not only tend to fill up their containers, but they also attempt to escape from containers as rapidly as possible when they're unsealed. So there's just a little bit of refresher on what's in the air that we breathe. So if we want to test the air we breathe for pollutants that are caused by emissions like from cars or from industry or power plants or things like that, it's important that we recognize that we can't test all the available air. There's too much of it. Trying to contain it in any way is pretty much impossible. What we can do, though, is we can take small samples of the air we breathe and we can put it through systems that are designed to test for the presence of pollutants. with think like even just a simple smoke detector. What it's designed to do is take samples of the air that are passing by, and then to examine that air for what are called ppm, parts per million, of pollutants. And then you can use that data to project the findings to the entire atmosphere within a certain radius. So a smoke detector, for example, if it's detecting smoke particles in a high enough proportion, it'll go off and it'll warn you, hey, in the immediate vicinity, there's a fire and you better get out of here. Air pollution type detectors can do very similar types of things. They can examine the ppm of different pollutants that might be in the air and provide data that say how polluted the air is and whether or not it's becoming dangerous to breathe. So with a very small sample, we can have a good understanding of the composition of the air that we cannot see, we cannot contain, but which we need to breathe to stay alive. And we can also, kind of a cool part of the science that's involved, we can take samples from different areas and we can make comparisons, which is really important if you want to see how pollution compares from city to city or nation to nation, or if you want to understand the effects of pollutants that are coming from a specific source. So, if like there's a volcano that goes off and you want to see how the volcanic ash is affecting the air, you can do that. So again, taking a sample of the air we breathe, think about how do you do that? It seems very difficult to do, but we're able to do it. And because we're able to do it, we're able to make sure that the air is safe for us to breathe. Well, let's think now about food safety. And as I've mentioned before, I was a McDonald's manager. That was one of the things that I did. And I actually got to do food safety tests every day. So let me ask you, how do you know that the food that you're eating at McDonald's or any other fast food restaurant is actually safe to eat? Most of the time, the food that you're going to eat at a restaurant or that's been packaged or anything that's been prepared hasn't gone through any sort of testing, because most of the time when you're doing food safety tests, you have to destroy the food once the test is completed, because by injecting probes into the food and things like that, you are contaminating the food, potentially. So you can't eat food that's been tested. That's a general rule. So what food safety inspectors have to do instead is that they have to take samples of food that have been batch processed, and then they have to put the samples through tests to ensure that what's on the way to the store isn't contaminated. So, for example, at McDonald's, we would cook 12 hamburgers at a time, or 12 hamburger patties at a time of the regular little hamburger patties. And we would put those in a tray and then use those when we were making sandwiches. And every now and then, I would have to come by and have them make me a tray of meat that was exactly like the tray of meat that they were using for production, that using the same settings, the same techniques, everything. And instead of that tray of meat going into a slot and being used to serve food, that tray of meat would instead be tested for food safety. I would use some different measurements to test it for food safety, and then I would dispose of it as waste. So the idea is that I was collecting burgers that were just like every other burger that were being out. They had been frozen the same way, they'd been processed the same way, they were being cooked the same way. So we had an idea of whether or not the food that we were preparing was safe or not. Now that in some ways should terrify you, because you might think, well, gee, what if there were a few burgers that they left sitting on the counter and they thawed and they got some bacteria in them and then they cooked them? Those might not hit the food safety test. And the answer is, you're right, they might not have. Hopefully, if that was a regular practice that somebody was doing, it would have gotten caught eventually and we would have been able to fix it. But if it was just a one-time thing, you're right, it might have slipped through. So food safety tests aren't 100% effective. And there are times, too, where tests generate a false positive or a false negative. And sometimes that causes catastrophic consequences that result in a lot of people getting sick. And by the way, that can be really true of all things. With vegetables, like spinach or lettuce or other leafy greens that are grown using fertilizers that are derived from the meat industry, because the pathogens can get in the fertilizer and they're harder to detect because they're on the surface of the food rather than within the food. And then there are other times where there are diseases that are not being screened for that get into the food supply, and the food safety tests just can't catch them because they're not designed to catch them. But the good news, generally speaking, those tests do work, and sampling the way that food is prepared does give you a pretty good indication on whether or not it's safe to eat or not. And again, the process it has to be sampling, because otherwise all of the food we eat would have to be destroyed. So sampling, it works for the most part, and most of the time it does prevent catastrophic consequences from occurring. Now, I want to point out all three of the techniques that I just talked about rely on one important idea, and that is that everything that we're going to test is randomly selected, so that the findings can be projected to the population. Okay, in the case of the blood test, we're getting a little bit of blood that's coming randomly from your bloodstream, and it's yielding findings about the entire bloodstream. In the case of pollution tests, the findings are from a little bit of air that's been selected randomly, and it can yield findings about the entire atmosphere within a certain radius. In food safety, findings from a few randomly selected pieces of food from a batch of hundreds, thousands, or tens of thousands can yield definitive insights as to how safe that entire batch is. Now, if we don't use a random process in our testing, the results can't be projected, and the findings can actually be quite dangerous. So randomness is important when it comes to sampling, and we need to understand when it is and is not appropriate to draw random samples. Well, now let's think about sampling as it pertains to human beings. And I think one way to think about it is that sampling allows us to talk to some people to say that we know what all people in a population think. Provided that, first of all, that the sample frame error isn't too high, that the sample is randomly selected, that the sampling method that we're using is appropriate for the population that we're studying, and that we don't introduce too much sampling error or non-sampling error, which again we're going to talk about in lecture series 7. So the idea here is we're taking samples so that we can talk about what a group of people have to say, and we can project that out to what all people in that group, in that larger group, that population, have to say. And that's really powerful and really important. And much like a blood test, pretty reliable if we do it correctly. The trick is in making sure that we do it correctly, and that's going to be the topic of our next lecture.